Hello, spider enthusiasts, spider friends. Today is a fantastic day. I am super excited. Um, I am joined today by the one and only Becky O'Neill. Becky, if you wouldn't mind, go ahead and introduce yourself.
Yeah, my name is Becky Hansis O'Neill, just Becky O'Neill for short, and I am a PhD candidate at the University of Missouri St. Louis. I study tarantulas and bumblebees. I do a little bit of everything. I do a little conservation work, but my big interests are animal behavior, and I'm really interested in sentience and invertebrates, like how do they process information? Can they feel things about that information they're processing? And then also, how do we measure what an invertebrate might be feeling? So those are my big research questions. And I also keep a pretty small collection of tarantulas. I think I've got 10 right now, including my lab spiders. So yeah.
What species are you working with, Becky?
So in the lab, I've got a Phonopelma annex, and then Brachypelma bomi, and a Phonopelma simani in the lab. And then at home, I have a Pamphobetus machala, Carabina versicolor, just got a green bottle blue, and then I have a couple curly hairs that I inherited from other students, and they're kind of pet holes. They're very happy to be in the burrows.
Yeah, that's a good thing. If they're a pet hole, it's usually a happy spider. So you're working with spiderlings, or do you have all life stages?
Right now, I just have juveniles and up, but my versicolor I raised from a sling, Pamphobetus I raised from a sling. The bomi was a subadult juvenile when I got him. Yeah, and at this point, I'm usually getting slings unless something's like coming in for a research project that's already been an adult. But just for me personally, I like to get slings. I feel like it's not always perfect, but there's less of a chance that that's a wild caught animal. And I learn a lot raising the babies.
Absolutely. Yeah, I agree with you there. That's how we like to do it historically when we started. First of all, guys, let me back up. Let me back up and introduce myself. My name is Ryan. For those of you who don't know me, I am the owner of Marshall Arachnids. Myself and my partner Jessica operate a small business out of our home where we breed tarantulas for the pet trade. We do a little bit of dry goods here and there. If you don't know our background, Jessica and I are ex-zookeepers, so we have a zoological background. Jessica has the piece of paper that says I am a zoologist. Me, I just got lucky and had to work really hard. But we have somewhat of a biology background, and this is how we kind of come to fancy Becky's work. So we actually, Jessica and I discovered you on Richard's podcast, the Exotic Pet Collective, I believe is what he's coined that one. And you were on discussing invertebrate memory, right? How they were capable, how they may be capable of learning, right?
Yeah, learning and cognition, and they definitely learn. So there's a lot of cool arachnid research in that arena. But yeah,
yes,
and that was totally fun. I love doing that.
Yes, Richard is such a good host. Yeah, he is fantastic, and he asks a lot of great questions. And then fast forward, you had discovered that I was on his podcast talking about thermoregulatory behaviors and our observation with our collection of animals. And in response to that, you decided to write a blog post, right? Which you then shared to the Facebook group Tarantula Collective, correct?
Yep.
Is that how that worked?
That's how it went down.
Perfect. And in that blog post, which I actually just might have to come help me with I have it pulled up. Let me see if I can find it here.
Oh yeah, that's it.
There it is.
That's my new Carabina versicolor. All grown up with
Look at that.
Yeah, that's the one that I provided just a little extra heat to, and he's a wonderful eater and Weber, and
amazing. Yeah, that's a healthy little animal there. So, and again, you've raised this one since a spiderling, right?
Yes.
Okay. Yeah, so that's the post. And in that post, you had mentioned too that you started with a versicolor, and then you lost that animal, and you kind of tried to troubleshoot what might have gone wrong with it, right? So then you made some adjustments, and you provided it with maybe a little bit of supplemental heat, and here we are today, right? You've got that animal, and we're doing well.
He's on his way to maturity. Might have to find him a lady friend when he hooks out.
Yeah, we can certainly help you with that. But I find that really interesting. So Caribbean versicolor is one of our favorite species too. And, in the hobby, the animal in particular has a reputation for being fragile and delicate. And what caught my eye about this, above all else, I mean, was really that you made a change. You provided the animal with heat, and now here we are. Is that the only parameter that you changed with that spider?
Let me think through I'm thinking of Caribbean reverse color number one, what I did with him. So the
Yeah.
first spider I had a little bit of heat just by accident, the one that died while it was growing.
Okay.
And that's because they're down in my room. I have a few reptiles. And where I put this cage, there was just happened to be a warm site and a cool site. So the spiderling got pretty big, and I said, okay, time for a rehouse. And also, you're so beautiful. I'd like to look at you a little more. Why don't you come live on my desk? And after 2-3 months, I found him still stuck to the wall. Abdomen looked great, but this was a dead spider.
Okay.
Definitely like floppy, not, and I took him and I'm like, okay, well, if it's an ectotherm, it's not dead unless it's warm and dead. So
Right.
just confirmed. And then after that, I said, you know what? The only thing I can really think of that I changed was lost heat. So now for this new versicolor that's gotten much larger, they're staying down in the reptile room. They actually have their own heat lamp, and they're able to thermoregulate and do their thing. And they're a super good eater, lovely spider.
Perfect. And you've obviously like monitored the temperatures that they're exposed to, right? I think I saw that in your blog. You're aware of what gradient you're giving them, right, from day to night or whatnot.
Yeah, I monitor it a little bit less than I used to, so I could run down there with the heat gun and be like, what are we at right now? But basically, I'm making sure, like, the spider shouldn't be any more than what I would consider warm on my hand. Mm-hmm. Like, there shouldn't ever be hot, and it should only be a little warm. Like, so I'm making sure, and they need to have refuges in their tank that are much cooler. So
yes,
I'm not down there all the time with like my temperature gun, but they're doing how they are.
Yeah, that would be exhausting. Yeah. Yeah. Okay. Interesting. So to kind of back up a little bit too, for those of you who know Jessica and I, we have been almost annoyingly so like proposing this idea that the animals like the tarantulas in particular, we're just focusing on those guys for this podcast, are indeed like poikilothermic, meaning they need to thermoregulate via basking and burrowing. And that has always been kind of at the forefront of what we do, right? I think like that's the first question I always ask customers is how warm is your space? Are you willing to provide it with supplemental heating? Have you thought about maybe how you might do that? And unfortunately, a lot of times we're kind of met with like, what are you talking about? You know what I mean? Like a spider, they're fine if you're comfortable. That they're comfortable. That you guys have all heard that before. And it has historically created kind of a point of contention, right? And it's always kind of been a point of contention in the hobby itself. But with us, with the amount of animals we produce, and how we kind of keep everything in house, it's very important to us that this message is kind of well received with the animals that we're sending out. And I would say, it's getting better, but people like Becky who have the credentials, have this scientific approach to this stuff are, I think, critical to the hobby in the sense that they can help us kind of break down that wall. To me, like to kind of paint a picture here, what's happened is in the hobby, we've kind of hit a brick wall, right? And for the longest time, we're in this car, we've been stuck at the brick wall. There's nothing beyond the brick wall. This is it. This is how we do it, right? But as animal science has caught up, now we know like, oh, there's some light coming through the wall, or oh, there's a little keyhole I can see through the wall. Oh, maybe the wall ends. Maybe I can go around the wall. It's kind of dramatic, but what I'm seeing is the gears are starting to turn. We're starting to catch up. We're starting to understand that there's a lot more complexity to these invertebrates than once thought. In fact, I am so bold to say that I think we've kind of known this since the 70s that the animals have thermal preferences and whatnot, but it just hasn't made its way through the hobby. But now, in light of the current climate, starting to come through. More people in academia are keeping these animals, like Becky, and so this is an exciting time to where we can kind of all come together and start changing how we perceive things and how we apply the husbandry, because also the technology and the tools available to us have gotten better, right? It's easier now. It's easier to provide these animals with these little things and these little microclimates that they capitalize on, right? So it's very exciting. I went off on a little tangent, and Becky, I do that, by the way, if you want to just like come in. Otherwise I just keep going.
No, I have thoughts on the brick wall you're talking about. So even in the 70s, when scientists were doing this kind of work, they still weren't thinking about welfare and invertebrates. I guarantee you nobody was adding heat, even though they knew that these are animals that were thermoregulating. And I think what we're starting to see really in the past 5 to 10 years is interests in invertebrate welfare across the board. So like Canada now has regulations for how you work with octopus and other cephalopods. In the past year, there's a new group called the Insect Welfare Research Society that's all about welfare and sentience. And they include tarantulas. Even though their big focus is insects, they support research on everything. And what I think you'll find is there's also still debate in the scientific community on what kind of welfare invertebrates need, what kind fish needs. So getting away from all the cute animals and the non-cute ones. So I think in both the science, the zoo world, and the hobby world, we're in this zeitgeist right now of really figuring out what the heck does good invertebrate welfare look like. So I'm hoping those are the cracks in the wall that you're talking about.
Yes, absolutely. It's like there might be a little more to the story, and I kind of want to break myself down a little bit because I've come off, I think, a little. I don't want to say the word aggressive, but I've been very adamant about this, and I think it's rubbed a few people the wrong way, and I do understand that, and I know that. But the problem is that your experience is your experience is your experience, right? You only know what you know, and you don't know what you don't know, right? And I think the biggest thing here is like I'm coming from the zoo world. I've worked with welfare coordinators. I see how that is all being quantified, right? I've also done some conservation myself, which we can talk to a little bit. I've worked with amphibians in Honduras on a very neat project, which is still ongoing. I'm not part of the crew anymore, but I still know all about it. I know how it's working. But what I learned is like spending some time in nature watching these animals too. Like you don't have to be a scientist. You don't have to be anything. You just have to spend time outside watching what happens in these unique places, right? And it's like there's so much more going on, and it's not black and white. There's so much nuance and detail and all of these little things that kind of come together that make up what we're calling welfare, right? And I want you, Becky, if you wouldn't mind, in a second here, let's talk about the 5 freedoms and kind of like how that might play into invertebrates. Because a lot of people are like, "What are we talking about here? 5 freedoms, welfare. What is this?" Right? So let's touch on that, how that might play a role in like our animals that we're keeping in our enclosures, like you can see behind me. What are the 5 freedoms of welfare?
Yeah, and I don't remember all the specifics of the history, but the 5 freedoms or the 5 domains, which are slightly different, have been around for over 20, 25 years. And they came out of, there's a particular guy, I think, that wrote about this and has done all the revisions, but basically ways that we can try to measure good welfare outcomes. The big thing with the 5 freedoms is they're usually freedoms from some sort of negative state. So they're kind of like rules. So the first one is the animal should be free from hunger and thirst. The second is they should be free from discomfort. The third is free from pain, injury, and disease. The fourth is they need to be free to express their normal behavior. And 5, freedom from fear and distress. Too many F's in that sentence. The difference when we get into the 5 domain model is that, okay, you're talking in the 5 freedoms about freedoms from all these bad things happening. The 5 domains is like, okay, how can we also increase the good things that are happening to an animal? Not just making sure they're free from bad things, but giving them opportunities to experience good things. So instead of calling them the 5 freedoms, they're now the domains. So you have the domain of hunger and thirst. So a negative would be like, if you're hungry or thirsty, that sucks. A positive would be giving you a lot of variety in your diet. You're not eating the same thing every day. The opportunity to have a really positive experience around feeding and stuff like that. So those are 5 freedoms. They're super, they come up a lot in zoological facilities, animal shelters and rescues. I'm even going to say the meat and food industry, even though that's pretty complicated.
Yeah. Yeah.
And in research as well. So I don't know if folks know this, but if you're doing animal research at a university in the United States, you have to go through a bunch of approval procedures to make sure you're not causing undue stress and harm. However, you don't have to do that for invertebrates at all. If you want to do a study on tarantulas about pulling their legs off, nobody can stop you legally.
Interesting. Yeah, that's interesting. I didn't know that actually. Maybe I did, maybe I didn't. I don't think I did. But so everything you just talked about, it's really interesting. I think a lot of people would be like, okay, but how does that, and this is, I can speak from experience. This is still an issue in AZA, which is most of what you just talked about is really easy to apply and quantify when you're talking about mammals, right?
Yep.
Talking about lemurs or lions. It's very easy to go through this little checklist and quantify these things. It's so much easier than working with an invertebrate or any ectotherm, really, reptile, amphibian, right? So with that, I think it's still what I'm trying to say is it's still an issue in AZA, right? It's like how do we tailor that to the different taxa, right? And the hobby, of course, it's no exception. It's like, okay, great. How do we quantify and measure these things so that we're doing this successfully with our invertebrates? And one thing that myself and my other colleagues at the zoo would always try to say is like, well, let's look at natural history. Natural history tells us a lot about how we can measure those things, and with that comes behavior, right? So what does the animal do in nature, and how do we help facilitate those behaviors in captivity, right? So does that make sense to you, Becky?
Yeah, you're really focusing on freedom number 4, which is the freedom to express normal behavior. And then the animal can really pick for itself what is comfortable and what is not comfortable and keep itself from being hungry and thirsty. So yeah, giving them the freedom to behave, I think, is a huge factor in good welfare.
Perfect. Yeah. Yeah. And I'm kind of blending all these together because to me, it is all one cohesive thing. You can certainly go through the checklist, but that was where I always struggled as a zookeeper. I'm like, it's all just one thing. You know what I mean? You can kind of chuck them off one by one, but it's like giving them the freedoms and giving them these resources to choose. And that's something I've been talking about too, is giving the animals the ability to choose, especially when it comes to thermoregulatory behaviors, right? Letting them warm up a little bit, letting them cool down a little bit. I like this general rule of thumb that you kind of just talked about, which was like, it shouldn't be hot to my hand. That's great. I've never thought of that. Because I'm always like quantifying with numbers. I'm like, give them this number or roughly, you know what I mean?
Yeah.
But I like that just that's everyone can hold their hand under something and be like, that's too warm. You know what I mean? Or that's too cool or whatnot, right? But also the problem too that I've come across is that like it's kind of subjective, right? Like, and I've always thought that too. I'm not very happy in temperatures below 70 degrees Fahrenheit. Like, to be honest with you, 75. I don't like it. I'm not comfortable. But a lot of people are fine in 65 degrees, right? So you can kind of see where I'm going with this, where we're kind of getting away from A, natural history, and B, like this quantify, like we're just getting further away from that. We're like, well, I feel good, right? We're kind of dismissing the animal's behavior, the natural history. It's like I'm good, so they're good, right?
Yeah, exactly. And I've noticed anytime I've gone into an invertebrate facility at a zoo or anywhere else, non-zoo, where just someone has a lot of them, usually, especially if they're good keepers, they're keeping that room much warmer and more humid than all their other rooms. So even if they're not giving individual heat to individual animals, it's not like your house, it's warmer.
Yes, it's uncomfortable. Like, and I'll be honest, like I've walked in because I worked for Omaha's Henry Doily Zoo, and they have a beautiful insect butterfly pavilion. And I know when I walk around and back there, I'm like, I take some clothes off. Like I'm a little warm. I work with herps too, so it was always warm. But that's something very easily like when you walk into these places, you're like, it gives you the feeling of like you're an immersive rainforest experience, which is not typically in most homes what you're going to feel right in your bedroom or on your desk.
Yeah.
So that's a very simple, basic way to be like, there's a difference here. We're talking about something else. We're talking about an ectotherm, and maybe we want to turn it up a little bit, But the opposite can be true too, which is like, that's why I always say it's measure, because that's where if you start applying heating elements to your animals, you can go the opposite end, right? Which is just as dangerous. And that's where like, oh, they cook themselves. They're not capable of thermoregulation. I don't know the history of it, but I guarantee you a whoopsie is what happened. And that's how this stuck with a lot of tarantulas,
And the way I get around that, because, what if I accidentally put the wrong wattage LED in my I like those little dome heat lamps. Those are my favorite.
Me too. Me too.
What if I get something wrong? What if something changes in the habitat in my room where I keep everybody? What if it's warmer or cooler than I was anticipating? So one of the things I've started doing is always giving it cool burrow. So if I screw something up, that animal has somewhere to go. So that if I make a mistake, it's much less likely to lead to the death of that animal. Hopefully I'm going to be able to catch that in time. So for me, it's like, you got to have another burrow for a retreat.
Yes. And so I'm so glad that you mentioned that, because a lot of we keep animals in very large enclosures. Behind me are 12 by 12 by 18, which I would say most people say is a little bit too large for the animals. I disagree. In fact, if I could go bigger, I would.
My Panthervatus is in a 40-gallon breeder.
Perfect.
She's a big tank.
Yes. And they might not utilize all of that space, but they have the option to do so and get away from a whoopsie. Say you're like, okay, this thermoregulatory behavior thing makes sense to me. I'd like to start experimenting. Well, you've created a safeguard for yourself and for your animal, right? Giving them that space. Just a little something I wanted to mention, because you mentioned a good point. When these things come up, I got to jump on them. So more space isn't necessarily a bad thing, right? I know some people who are successful reproducing these animals in deli cups, right? Something this big just by heating the room. And Becky, I want to kind of ask your opinion on that too. Like, you and I are kind of on the same page about providing these gradients and whatnot, but how would you respond if someone's like, well, I keep them in a 128-ounce deli cup and I just heat the room and it's fine?
Yeah, I mean, we know physically that there's a range where they're going to be fine. So alive does not mean great welfare, would be my response. So there's animals where you could probably keep them their entire life in a setup like that, and they're going to live. But we don't have enough information on, is that animal suffering or not? That's the Wild West of research in invertebrates right now, is how do you measure suffering? Big debate. Also, we don't know, would your animal have grown a little bit bigger or lived a bit longer if it was in a more naturalistic setup? And there's studies, like it was in my other blog, but there was a good one, and I want to say it was either pink toes or versicolor, I forget which genus, but they raised spiderlings in enriched and non-enriched enclosures, and then they tested them. They had like a little field and filmed them. And the non-enriched tarantulas ran away. They were more likely to run away further, run away faster. And they were much more likely to display certain defensive behaviors. So to me, that tells me, okay, they were alive and fine in their deli cups, but when compared to animals in a different kind of enclosure, that to me, from a zoo perspective, looks like chronic stress, right?
Correct.
Those kind of
I'm glad you said that. Yeah. So we're looking at a behavior, right? We're looking at known behaviors, and how they react when given something larger, right? Or a bigger, better retreat or something like that. And I kind of want to circle back around too, if you wouldn't mind. And with Richard, you had mentioned that study where they tried to measure cortisol levels, right?
Yeah.
I'm familiar with that study. Actually, right when I left AZA, one of the welfare coordinators at the zoo I was working at sent me that study, and I was able to check it all out, and I was familiar with what you were talking about. I kind of picked that apart, and I kind of want to talk to you about that.
Sure, because it has there's some big pros and cons in that study for
big time.
Yeah.
So just from my perspective, I'll share with you. The pro was we can measure stress levels in some quantifiable way. Boom.
Cortisol and arachnids. Who knew?
You're right. Amazing. Yes. So
Oh, that's great.
they can be stressed. Yes. Boom. Okay. But moving forward with that study, this is why someone needs to do this again and
Yes.
multiple times over. But I'm all about UVB, and because we're big proponents of full spectrum lighting, and I provide, if you can see behind me, I've got some different flavors of color temperatures and whatnot. But I'm all about providing them with full spectrum lighting and even UVB in some regard. Now, I haven't gone down that
Yeah.
rabbit hole, but what I noticed in that study is I think they were trying to measure when giving them UVB, like what is their cortisol level, right?
Yeah. And they blasted them. Those lights were really close to those animals. And they have a few sentences with an OBT, and they were like, yeah, this animal just kind of went nuts. So
Yeah, yeah. They were shoe boxes, right? They were something small. You kind of lift the
yeah.
lid off.
And
Yeah.
if
So
that researcher is out listening, I'm not like, I am so happy you did this work. Please don't see this as like a personal.
so am I. But it's important.
Yeah.
I want to talk about something too, because as another keeper, someone who is familiar with tarantula, natural history and husbandry, there are a lot of factors in that study that could have also, more so than the UV, contributed to the stress levels in the animals that were being tested, right? Like, and I don't think there was very much soil for the animals to behave naturally.
No.
I thought, if this is the same study, there was a heating element provided underneath those bins. Am I talking about the same or something different?
I don't remember, because the last time I read it, I wasn't thinking about thermoregulation, so I'm not sure.
Okay.
And I kind of read that study as, we want to do some things that are going to stress these animals out without killing them, so that we can actually see this hormonal change if it exists.
Yes. Okay.
And it was also a very small amount of animals. So in terms of like what cortisol means for invertebrates, you were totally right. We got to do way more studies to figure this out. But yeah, I would not base any husbandry off that study. I think they were trying to get them stressed out to see if the cortisol would even work as a measurement.
Yep. So there's our pro. Yeah. I just wanted to touch base on that because now it's out there that it was done. And as a keeper, I just wanted to point out there were a lot of factors that, like, I think we should just redo that with some input from some other keepers, perhaps. And then because I'm very interested to know exactly what UVB is doing to these animals, because we know they have a low to moderate exposure to it in nature. We know they can regulate their exposure to it in nature, right? They can get away from it, right? So I'm very curious to know what benefits maybe UV has on them, which is now we're getting in the weeds, you know what I mean? But it's still part of natural history. It really is. So,
and I've thought about that, and I don't know as much about like vitamin D metabolism in invertebrates, because we know that invertebrates, we see changes in values when we run blood tests and things if they get UVB. Then we also know if you get too much UVB, you get cancer. So so it's touch and go. I think the UVB argument is way more complicated, I think.
I know we are getting into it. And we're not even talking about airflow and moisture and humidity. We're just talking. I know there's so much to this, but
what I would like to know more with tarantulas and UVB is photoperiod, especially if you're trying to breed them. Like, that could be where lighting.
I can comment on a little bit of that. Yeah. So I obviously use full spectrum lighting. You can see I've got two flavors behind me. One's kind of got a pink hue to it, and my thought was maybe we'll see them more. I see my animals every morning. It doesn't matter. The lights are irrelevant, because if you notice, I don't think you can see, but I've got large cork hollows that every animal can get away from the light completely, right? But every morning they come out and bask. And I was thinking, well, what if the red flavored light, I would see them more frequently, and no difference. Like on it, I see them the same. They come out, they get their morning cup of coffee, and then they retreat back to their hollows. But what I will say is how dialed in these animals are to like the circadian rhythm and photoperiod and like the change of seasons. Like they are so in tune with that. And the way we've been able to get away with it is we pay attention to when the sun starts to set, and we turn our lights off an hour before the sun completely sets. So they have that dusk period. And Jessica and I can count the seconds before every animal comes out, every single one. You know what I mean? So like they're really cued in to photoperiod and the changes, and we've been able to dial in. We've found that breeding them in the fall when it's kind of cooler and at least here it's really humid, and then they all go nuts and lay egg sacs in the spring. right? When it starts to get warmer and the days get longer and there's more exposure to the sun and all that stuff, right? So this is not scientific, but I'm telling you right now, big difference. They are cued in on those changes for sure. It's very interesting. Yeah,
We
they
had
they
a
kind of
power
go
outage
ahead.
at the lab and they all were just having a party. Even though their rhythms were messed up, they were out and about and like, all right, it's dark. It's
Isn't
time.
that yeah, yeah. So there's some, I think from that you can gather there's some kind of leeway here, right? Like they're gonna cue in on the change no matter what. And you got to assume too, lights go out. To me, it's a sensory cue that like humidity is gonna spike, temperature is going to fall, right? It's a cue. It's like it's time to be a spider. So get up and get at them. You know what I mean? Yeah, they're
Yeah.
all kind of really cued in on that. Yeah. Which is
And
why I think photoperiod is just as crucial in my opinion, as giving them heat and water and food. To me, it's all kind of like same thing, you know?
yeah,
Yeah.
and I've admittedly been a little lazy on my photo period. I keep everybody on a 12-12 schedule for my
Yeah.
own sanity. But if I were going to be
Yes.
breeding, I don't think I would do
Mhm.
that. And I've even thought to myself, like, well, if I move and I get them in a room with big windows, maybe that would be better for them. So this is something I've thought about and just haven't done a lot with.
Okay.
And I can also
Yeah.
say for the wild tarantulas that I study, Aphonopelma hensi,
Mhm.
they are super in the wild. They're really consistent at our field sites about when they're laying egg sacs, when we see the
Right.
males, when the first ones come out, when we start seeing less of them for the year. They're very, very strict. And I've thought, is this a temperature thing or is this a photo period thing or both? And
I bet it's both.
I
Yeah.
haven't read
I,
enough about it.
yeah. I tend to err on the side of like, oh, it's temperature related. Jessica's all about photo. That's what it's all about. Yeah, because all of her field work with, I think, she's over my shoulder here. Box turtles is what you were working with.
Ah,
Yeah, so she's like, yeah, photo period is everything. Yeah, it's like they really cue in on that.
yeah, and
But that, yeah,
maybe we should back
go
up
ahead.
just for a second and explain what the heck we mean by photo period.
Thank you. Yes, please, please.
Because yeah,
Good call.
if
Yes.
you're biology nerds, you're just like, woohoo, we're off to the races. So
Yeah.
photo period is going to be the amount of time an animal has light, and if we're talking about wild animals, that's going to change through summer, winter. Um, for some animals, like if you're at the bottom of a rainforest floor and you're near the equator, maybe that's not such a great cue for you to do things. But basically, many animals use the amount of light that's happening each day to regulate different aspects of their biology, like reproduction, brumation, stuff like that. So that's my 2-minute elevator. What is photoperiod speech?
Thank you. Yeah. No, this is exactly why I wanted to talk to you. Yeah, you got to catch these things and break these things down and make it easier to understand. Otherwise, and I think the biggest takeaway here, guys, is like you don't have. We're not saying like just change everything all of a sudden all at once,
Yeah.
but it's to start thinking. There's a little more going on, right? They're cued into environmental, um, I guess cues, I guess, right? So it does make a difference in all this is all about welfare, right? It all kind of comes together to make happy spiders in captivity, right? So, but good call. Yeah, definitely break that down. I wanted you to mention, you said in your blog you went to Costa Rica. If we
Yeah.
could touch on that. I'm envious. I'd like to know more.
So I'm really lucky that the University of Missouri St. Louis is part of a group of organizations that are part of a Costa Rican organization called the Organization
Nice.
for Tropical Studies. So I was allowed to go down there for college credit for 6 weeks this past summer
Awesome.
to work with scientists from Central and South America and work on a whole bunch of different projects. Um,
Okay.
I saw whip spiders. I saw a tarantula. I was so busy. This was like 13-hour days that I
Yeah,
wasn't
I bet.
out there all the time looking for tarantulas. But
Right.
I got one of my rooms at one of our field stations right outside my door. The biggest Aphonopelma semani I've ever seen had a burrow. And everybody
Ooh.
loved her. They started in the class like giving her little bugs whenever they went by. So we probably made her kind of fat.
Yeah, yeah.
Um, but yeah, so I went down there. I got to work with lots of different scientists. I got to study Apeliones, so daddy longlegs for common name. Did a lot of work with frogs, orchid bees, which are beautiful bees if you've never seen them, and then
Ooh.
water bears too. So I got to do all these little
Oh,
science projects. It was so fun.
that's amazing. Yeah. So what regions, where were you at? Where were you studying? Were you all over the place, or did you focus in one spot?
So we moved about every couple weeks. So we had
Okay.
lowland rainforest, we stayed in cloud forest, we stayed in dry forest, and then there was at least one other kind of rainforest. But the geology of Costa Rica and just where it sits in planet Earth essentially mean that there's tons of these highly biodiverse little ecosystems that are all just a little bit different. And that's one reason there's so much high biodiversity there, is there's so many different types of habitats and lifestyles that all these animals can have in one place. So super cool, super fun.
That is super cool. Yeah, and that's and when I saw that you went to Costa Rica, I was hoping that maybe you had went to a cloud forest and saw Mesomelis, Megaphobia Mesomelis with the red knees.
Yeah, I didn't see them.
Okay, are you familiar with them?
I'd actually have to look those ones up. Let me see.
Yeah, check them out. They're a gorgeous Megaphobia Mesomelis. They're kind of, for me, they did just chimed in, said they just got reclassified.
Oh, well, I was able to look them up with the old name. They're gorgeous.
They're stunning, and what's really interesting, they are in the hobby. I'm looking over at one right now, but it's really interesting. Their habitat fascinates me. Cloud forests fascinate me, and it kind of speaks to the whole thing that I've been saying is like a tarantula is not a tarantula is not a tarantula, because I would never treat Mesomelis the way I would treat a Brachypelma auratum, like another red knee tarantula, right? Completely different biomes, completely different. I mean, really everything other than the fact that they're both tarantulas, right? Cloud forests are really unique in the sense that it's like really cool. It's really cool and misty, but there's usually high UV exposure in a lot of places. Like sunbeams come through the trees and kind of heat things up, but the air is cold. Amazing.
We were at almost 10,000 feet when we were at the cloud forest, and we hiked all the way up to the I'm not going to say the right Spanish word, but there's like these no tree areas with succulents and things that look a little like cacti that aren't cacti. But on our way up there, I would see these pockets of sunlight in the forest, and you would just see tons of invertebrates in that pocket. And it got me thinking about all the layers in rainforests. So what kind of tarantulas do you have up in the trees that I'm just never going to see as a person on the ground? So crazy microhabitats.
I'm so glad you said that. Like, there's just, and this is the complexity that I'm always kind of rambling about, is like, man, I'm telling you, just from one side of a mountain to another, it's just completely different. And your approach to just going back to husbandry again, it's just completely reconsider. You know what I mean? Because it's just completely different habitat, but it's in the same region, right?
Yeah.
I've seen what you're talking about. You get up through like the cool misty cloud, and then it's like real high UV exposure, and everything kind of shrinks, and it's real tough and spiky, but it's up at the top of the mountain, the same mountain, but completely different biome. It's really cool.
And just seeing, like, we saw a lot of bromeliad spiders. So if people aren't familiar with those, they're like wandering spiders. So if you've seen anything in that family, that's what they look like. And there's a lot of tarantula-sized ones in Costa Rica. And just the habitats of the bromeliad plants and where they sit and live is just amazing.
We've got a few of those bromeliad species from Brazil, the tarantula species. They're my favorite. Or actually, in our living room, we're going to build an exhibit for them right out of the wall. It's just like a permanent thing. I know I've always wanted them. I can't wait. I'm really excited. But for that one, same thing. We gotta figure out, UV exposure. Potentially, we're gonna play with that, and the real bright light for the bromeliad, and how we're gonna provide heat down that way, like for those spiders, because I know they kind of come out of the axle and bask. It's like, so it's a whole new thing that I'm really excited about. But anyway, that was a tangent. Super exciting.
And it relates to some of the tarantulas I see hensi here in Missouri. So just by accident, I had a student this summer that said, can I write down whenever we see a reptile? Because we're not studying reptiles. We don't really handle them unless it's for their safety, to make sure that they're not going to get smushed or something like that. So he wrote down all the reptiles, and then he threw it on a map with the tarantulas. And the tarantulas are super picky. They have little spots in our sites that we call tarantula towns. The reptiles and the amphibians are not. They have a very even distribution across this landscape, and the tarantulas are super picky. And what we're figuring out is they're probably picking the warm spots. So I have another student that's this summer more interested in microclimate kind of work and habitat disturbance that she's going to work on. So that tells me, because our tarantulas, if they're arboreal and they're making a web, or they're fossorial or terrestrial and they have a burrow, whether that's a complicated burrow or just a scrape burrow, they're really picky about where they put them, I think.
Yeah, that's very interesting. And I mean, that really kind of coincides with what I've noticed too. And it's all about, to me, like, at least, man, I swear, I haven't been out in the field studying these things, but I swear it doesn't matter what you give them. If you give them that little warm spot, that's what matters. That's the spot. I'm telling.
Yeah.
And not every species is the same. So you talked about Xenesthus, right? You have a Xenesthus or a Pamphos, right?
Pamphobatus, yeah.
Pamphos. Okay, so I'm noticing with those, they don't behave the same way that the arboreal animals do, or even some of the different terrestrial species similar to them. They're more like a dart frog in the sense where it's like the environment is the environment. Like they don't actively seek out heat source like the other ones do, which is very interesting. I mean, maybe someone else's experience might vary, but I found that interesting about them. And then now that tells me it's like another rabbit hole to go down. It's like, okay, well, what is different about where they're at that they don't do that, right? Like a typical poikilotherm, at least in my experience, right? What's that about?
Well,
they're just kind of out,
thinking about them and just walking around rainforests in Costa Rica got me thinking about this, that if you're in a rainforest near the equator, that's a super stable environment. So if you're a burrowing tarantula or just terrestrial, you probably don't have to devote a lot of your day to thermoregulation. But as keepers, we still need to have some ideas. Has anyone gone out to Ecuador and measured this? Because I have Pamphobatus machala, that's not even a truly described species. No, has anyone gone out and measured the temperatures that she's going to be most happy at?
Yeah,
so
I know.
So even for tarantulas that aren't doing a ton of thermoregulating, we still might need to give them options in captivity because we just don't have enough information.
Yes, 100%. And that's where with those species, I've always kind of taken the aquarium approach, something long, because I'm basically throwing behavior is how I just kind of judge everything. What are they doing? What are they responding to? What are they like? Okay, that works. Let's kind of work with that. But for those guys, it's like, what do you want? Like, here's this big range. Show me something. And it's just kind of like a shoulder shrug. I'd say, okay, all right, well, we'll see when you get a little older how you respond to, reproduction. I learn a lot through reproduction and how they respond to because everything kind of changes a little bit, when you start introducing reproduction into the mix as opposed to just day-to-day husbandry. So maybe I'll figure out something there with those, because I like them. I don't keep a lot of terrestrial species, but I really like the Xenesthus and Pamphos. They have cool behaviors, and they're not aggressive by any means, but they're certainly responsive. And I like their feeding patterns, their feeding behaviors. It's like really cool. Yeah. So
mine will come out if she's kind of hungry, I think. And
they definitely let you know when they're hungry. At least there's that. It's like, okay, I got you.
I have her regular feeding schedule, but once in a while I'll see her there and I'm like, maybe it's big roach time. Here you go. And she's usually pretty aggressive about eating at those times. So a lot of it for me is like watching the animal and responding to what they do. And I give her 3 burrows because she's in that 40-gallon breeder, and she stays in her warm burrow, which might just tell you something about, because she's on my lower shelf, it might just tell you something about that microclimate, like, oh, maybe I need to, depending on the time of year, add just more heat in that room for the animals that are lower. But she's also pretty active. Like I put cameras in her enclosure and she'll do laps at night. So I don't know. She's a funny one.
Yeah, guys. I use Wyze, WYZE. Wyze cameras are really cheap, inexpensive. You can put them on USB. They have night vision capability. That's something cool to see too, is what your animals are doing at night, especially if there's a temperature drop in the room. See how they respond. Wyze cams really help with that. Yeah.
This is something I've noticed with a couple of the tarantulas in our lab because I filmed them all night a few times. And this is only 5 animals. This is not enough to make scientific conclusions. But I notice some of them do laps. They're very slow laps because they're a tarantula, but all night laps. I think they were in a 10 or 15 gallon when I was filming. And that to me is strikingly similar to some vertebrate behaviors when they're captive. Not enough research to know if this is truly stereotype behavior like what we see. And when I say stereotype behavior, I'm talking about like if anyone's been to the zoo and you've seen like the big cats pacing, or I've seen it a lot with bears where they're going to do head swings repetitively. Um, no idea if tarantulas do this, but I am seeing if when I was doing that filming, some essentially, repetitive behaviors. But again, we
Yeah.
don't have enough science to know. Might not be stereotypy at all.
Right. They're just going places. Yeah, they're just, here's my space. I'm just gonna move. I don't, you're
Yeah.
right. I don't know. That's really interesting. I know that's usually a concern for new keepers. Like they put a new spider in a new environment and the lap thing kind of happens and
Mhm.
stresses them out and usually stresses us out. Usually it's just kind of like just let them give them somewhere to go. They'll find
Yep.
it eventually and it will be okay. But it's interesting. I've noticed that with my Zenesthus too, is they will do their laps. I've also noticed with that genus, they like to take away plants. They like to like cut down plants. They'll get a new growth and they take it away. They get new growth and cut it away. It's like stop doing that. But I wonder why. I wonder what that's all about. It's just
Yeah.
changing their environment. I'm not really sure, but
And that
they seem to like to do that.
goes back
Yeah.
to like rainforests. I don't know. And there's a genetics researcher, I don't have permission to like talk about it, so I won't, but she sent me a video of her pantho pulling up, like pushing on this bromeliad. Like, I don't want this.
Yeah. What
So
is that?
they do
Yeah,
know how
that's
to
very.
landscaping.
Yeah, yeah, they're just kind of making it their thing. I'm like, it would look a lot nicer if you wouldn't do that. But whatever. I mean, if to me, it's enrichment of some sort, right?
Yeah.
There's something. I mean, I don't know what it is, but I just know they're doing it. The plants just grow on their own. It's not like I'm taking it time and energy to like fix it or give that to them. They're just doing it. I haven't experimented with the ping pong ball thing with any of our animals. I know that's always kind of been historically kind of a topic of debate, but I'm with you. I saw your response to that, and I think there's definitely something tactile going on there. I think it's an environmental change, right? And they're just kind of checking it out. Would you agree with that?
Yeah, I don't buy this idea that, oh, maybe they think it's an egg sac. I'm like, I think that looks like an egg sac to a person. I don't think that looks like an egg
Correct.
sac to a tarantula, just based on
Yeah.
their sensory systems. That's not going to feel like an egg
Yes.
sac. It's not going to smell like an egg sac. So yeah,
Right.
I'm thinking there's this novel thing here. I'm going to check this out, and then they go through this sort of manipulation with their fangs and their feet.
Yeah. That I'm with you there too. I think it's just something new, something different. Because
Yeah.
they do that, right? They check things out. It's just, what else are they gonna do? They check things out. So
Yeah, and that would make a
interesting.
really cool study is to like, okay, let's give you some new objects. Let's see what you do with them. Let's do some
Yeah.
behavioral measures. See how you're,
And I've
yeah, like actually look at some behavioral outcomes for what happens when you provide that kind of enrichment.
Sure. Yeah. Yeah. I don't know what, as keepers we might gain from that other than it's just enriching, like you're checking that box. Like here's something novel, there's something new, and you can kind of maybe introduce different objects. But I've never seen arboreal animals do that. It's mostly terrestrial species, fossorial species that are interacting with those things. It's
Yeah.
interesting.
I tossed in
Yeah.
a ping pong ball with my hisser colony just to see, and the ball's never moved.
Yeah. Yeah.
So,
There's that too. Yeah. Yeah. You mentioned, about, I've heard this, I hear this a lot, that the animals are primitive and they don't have functional brains. And, the more research that we do, we know that that's incorrect too. There's more going on neurologically up there than what we thought historically.
yeah. So
Yeah.
if you're trying to compare a bird or mammal brain to a tarantula brain, you're comparing apples and oranges. And
Right. Something different.
huge parts of our brains are devoted to just to being able to sense all over our bodies. We're a relatively large mammal. It's one of the reasons you get bigger brain in a blue whale, but they haven't developed calculus or something. It's because their
Right.
bodies are really big, and you need more brain tissue to have sensory control and motor control over that body. So when you shrink an animal, like bee size, tarantula size, they're going to have little brains, and their brains are also differently structured.
Mhm.
The thing most closest to our cerebrum, our thinky part of our brain, they call it the protocerebrum in invertebrates. It's called the mushroom bodies. I have not
Okay.
found a really great picture of tarantula mushroom bodies, so I don't know where they're at. If you're looking for genius spiders, that's your jumpers. It's your jumping spiders.
Yeah.
So,
Yep. You're right.
yeah.
There's definitely a lot going on there. They're so visually acute. You can just tell by watching them. They're processing things a bit differently than
Oh,
your
yeah.
large bodied tarantula for sure.
Yeah, and I
Yeah.
think something just came out about individual recognition in jumpers. about how
Neat.
they can maybe recognize other jumping spiders within their territories. So they're
Oh, okay,
very
okay.
cool. But yeah, you can't
Yeah,
just say that a tarantula is completely instinctual. They have all the body parts and all the parts of their nervous system to allow them to learn. Um,
right.
I'm not going to go around and say they're like as smart as your dog or your cat or even a
Of
jumping
course not.
spider, but they're
Yeah.
totally learning about their environment, and the things you do within their environment affect what they learn and how they learn it. So
Yeah, yeah, I almost give them too much credit. I'm like, no, there's a lot going on there. Like they sense all these different vibrations, smells, and changes in atmospheric pressure, and like all these different things are going on in this little animal that on the outside appears to just be
totally.
inert, right? They're just sitting there, you know what I mean? Like waiting, being an ambush predator, yada yada. But no, I think there's a lot more going on than what anyone gives them credit for, and that kind of contributes to my attitude about it, which is like, no, there's more going on to your pet spider, which is why
Oh,
like
yeah.
I tend to really, really kind of I don't want to say the word shame, but the shoebox bone dry water dish approach to keeping something like that. I'm like, yes, they're fine. We know that they adapt. It's okay. But I think if you look, maybe if we all looked at them in the sense that like there's more going on, right? And they have got these little sensory organs all over their bodies. I mean, at the end of the day, I think really all they want to feel is they want their resources and they want to feel secure. But
Mhm.
definitely this whole topic of enrichment that you've kind of touched on with Richard and like we keep talking about like they respond to it, which I think that's the biggest thing is like quantifying that behavior is like what are they doing when they have access to these other things, I
Yeah,
think.
and
So
I think that
yeah,
would make a really cool citizen science project is to have people
I
do
agree.
observations on their tarantulas, they upload them, send them somewhere with some other information. Then you could get a really
Yes.
huge data set of lots of different
Yeah.
species to see how this is working. Yeah.
You know, I heard you mention that and like the kind of the undertaking that would entail like getting that
Yeah.
together. I've been bugging Jess about it if she wasn't so busy. I'm looking at her right now. I'm like that we could do that, you know, and she already is like the gears are turning. And
Yeah,
I wanted to kind of touch base on that too. We're kind of getting
sure.
close to an hour. I don't want to keep you too long, but moving forward, what is the future like for Becky? What do you want to do here? Like what is this
Yeah.
for to you?
So
What's future entail?
I want to get some more publications because right now, as a PhD student, without having published any of my work, I'm missing this sort of like, yes, other scientists reviewed my work and said, we're going to publish that. It's
Correct.
good. So for
Yeah.
me, getting this stuff published is really important. I know I mentioned to you that I'm just in the very beginning stages, and this is how I even got onto thermoregulation because behavior is more my
Mhm.
thing. Husbandry is.
Yes.
But I'd like to take the framework of the five freedoms or the five domains and apply that to tarantulas as an invertebrate model. And in a model in
Okay.
this case just means an example for here's how we can take traditional welfare ideas and apply them to an invertebrate because
Perfect.
in my opinion, we need to start doing that more. So that's one thing I'm working on. Want
Yes.
to finish the research. When I'm done, I'd love to start thinking about long term, and I don't know what this looks like yet. This is just a pipe dream.
Yep.
Can we have a proper tarantula society in the United States that's really focused on citizen science, conservation science, and bringing zoological institutions, keepers, and breeders and hobbyists together in a way that you might not see with other parts of the exotic pet community? Um,
Amazing.
that would be
Okay.
cool to me.
Yes, I'm with you. 100%. And you touched on something too, like we get this a lot. Um, how can it's kind of a double-edged sword. So a lot of people are like they're conservation minded. They're like, how can I help? Right? How can I do something to and I love that. Other people, you know, are kind of thinking like, well, we're conserving by being pet owners, which is not that's not really how it works in
No.
the world of conservation. You know, unfortunately not. Your heart and your mind are in the right place, but that's just unfortunately I think it's important for many keepers to acknowledge kind of the optics of how the pet trade is viewed by countries of origin and the CITES and all that. You know, it's
Yeah.
you know what I mean. But the truth is, the fact is that keepers can help out in a lot of ways indirectly, right? So like a lot of our husbandry practices can help out AZA institutions that have the know-how to start conservation initiatives, right?
Yep.
So that's one thing. Citizen science, like you said, if we can design the framework for that, is something else we can do. I think a lot of folks just being mindful of where their animals come from, and how they might have gotten that animal, and like understanding that process is also just knowing that, I think
Right.
would be great for about how the hobby kind of projects itself and people carry themselves, and talk online. I'm always doing the shh kind of thing, not because it's like, oh, don't do something bad and like keep it under wraps. It's just like, I love my spiders. I love them very much, and we do things very ethically, and we do to the best of our abilities. But I think we just need to kind of bring it in a little bit, you know what I mean, and kind of understand, like, with the current climate and welfare and all these things that are kind of coming into play in the agricultural communities and zoo and aquarium communities, like we are going to be under the microscope more than ever in the next 5 to 10 years. Period. I mean, that's just the way it works, right? It's just the way it's going. So I like what you're saying about changing the optics about how the hobby is viewed, right? And how pet keepers can actually contribute to this bigger picture of conservation. And
Yeah.
there are a lot. Go ahead.
I was gonna say there's some things that, because I think there's this idea that, oh, if we have all these captive bred spiders, and even we occasionally get some wild ones in, get some more genetic variation, that maybe we can release those someday. And we don't have good enough tracking for the most part as breeders within the hobby to know if this Carabineverse color is from the same lineage as this one over here. And you don't want to take a spider and release it in the wild if it has a bunch of genes from some other place, because they might not do as well there. So we have all these like issues. And what I'd love to have is a way, almost like a genetic barcoding project where you send in a molt from your spider, and we're starting to track how much genetic diversity is there. Can we see cases of hybridization in particular lines of spiders? Are we starting to get genes that we only see from some place in South America where they're not supposed to be coming from? So I'd love to see that. Again, it's another pipe dream.
Well, but it also, I want to take the time to kind of explain to the hobby too, that like everything you just mentioned with the genetic barcoding, like that's really how proper conservation starts, because you have to have a baseline. Yeah, exactly. You have to have that regulation, right? So, understanding that is important, but to like what you said, like there's so many animals in captivity, like getting that genetic barcoding started, and like having people aware of that process, and sending molts and like our animals for other experimentation, of course, moral, ethical experimentation, whatever that might look like, right? I want you to know, like, that might be on the horizon for hobbyists, to contribute in that way. And so, Becky, with as far as you're concerned, I want you to know too that like there are so many people that are conservation minded and want to help, but know they can't or don't know how. So like if you somehow find yourself in a position where money, like finances, because I know how expensive all this stuff is, if like we could bring the hobby together to contribute, even if it's just $1, right? And there's some incentive for hobbyists to contribute, like that is very possible. And that's something I want people in your position to know. Like if we could do that to raise funds for that, people would jump all over it, and vendors would help promote it and all that other stuff, because I think that is what is, in my opinion, going to save the hobby really, and not just the tarantula keeping sector, just keeping animals in general, right? I think that's going to be one of the only ways moving forward that like we're looked at differently and taken a little more seriously. And that's just a message. Like that's my pipe dream. I'm just letting now that I have you, it's like that. I would love for them to contribute. If nothing else, if not, financially, we could all just chip in $1. Every single pet keeper chipped in $1 for this sort of thing. Like it's all connected. Spiders, snakes, birds. It's all, you know what I mean? So.
Well, that gets into nonprofit management. Like, is this a sort of movement that could be led by a research professor at a university or a particular researcher at a zoo? Or should this be an independent nonprofit foundation at some point? Because we already have, for invertebrate conservation, we have, I'm going to say it wrong, sorry folks, Xerces Society. Like So is this an umbrella under something that already exists? So there's all these questions, and I'm like, I just need
Yes.
to get my PhD finished. So
Yeah. But I want you to keep that in mind
yeah.
too. Like if you ever reach that point where like funding is an issue, like I know like how where money goes and how where it comes from is again
Mhm.
optics, right? Optics are everything, you know. But once this ends too, there's a name I want to drop for you to see if you know this person,
Oh,
because they
really?
actually at our expo extended the invitation to me to send my stuff off for genetic testing to someone. And he dropped a number like $10 a molt, and we can start this process. I don't know if you know this person. We'll talk about it
Oh,
after the show. But
yeah, maybe.
yeah, it's and he has a government job working for USDA and Invertebrate pest control and whatnot. So
Okay.
maybe I can put you two together. I don't know.
Yeah.
Some people above me who are doing better, bigger, better things than I am. You know, if I could help in any way,
Yep.
shape, or form, I want you to know I'm on board. And I think a lot of people feel the same way. So
Sure.
because that's the only way, you know, we all grow together and we're all in it together. You know what I
Yep.
mean?
I'll keep it in
That,
mind for
that's where I'll leave
sure.
that. Yep. But it's difficult. There's these little things like that just kind of hold back progress, whether we
Yeah.
agree with it or how ridiculous it may or may not be. It's just that's just politics. So,
Right. And we could keep
um,
going. I know this is a little tangent, but I put a pin in it when you mentioned when we got into talking about captive breeding and making sure that the animals with the right genetic lineages are saved for conservation. Another
yes.
huge thing we don't know about are diseases in tarantulas. If you breed a
Hmm.
whole bunch in captivity that are part of a mixed collection and you let them go, what's the likelihood you're going to introduce a nematode or a virus to that wild population from your
Um,
collection? We don't know. We haven't,
yeah, you're right. There's no
like,
baseline. There's no, and I didn't
yeah,
even think about that. This is why I'm glad you brought that up. Yeah,
yeah,
that's another variable, right, of
yeah.
unknowns, which makes this whole conservation initiative like an issue. There's so many
Yeah.
hoops, right?
Yeah, it's
Um,
so hard. I don't think that means we shouldn't try
I
because
agree.
there are so many talented keepers and breeders around.
Yep.
Like to me, it
Yeah.
feels like untapped talent. But yeah, there's so many questions for how to merge our love of tarantulas and conservation together.
Yes. Yeah. Yep. I agree. And I think more conversations like this, putting conversations like this out there, kind of gets more people aware, whether they like it or not. You know, the conversation can start to shift and change, and these elements of welfare, which are inevitable, they're already affecting other industries, agricultural, zoo and aquarium science. You know, the hobby, I would say, is in some ways ahead, in some ways behind, and that's just simply because there's no welfare program regulating how keepers keep their animals, right?
Right.
But obviously, we all love our animals. We want to do the best for them. Um, and I just think that conversations like this and putting them out there is the one step in the right direction, making spreading awareness. And
Yeah,
um, anyway,
and that's why
is there anything else you want to add?
no, that's just what you said. Is that's why I'm totally open to having these conversations. Is because
Perfect.
yeah, we get this information out there just by talking about it more. And
Yep, putting it out there on the internet so that when somebody like a customer of mine Google's tarantula husbandry or tarantula care, you know, that there's these new 2023 upload, you know what I mean, that like people can start seeing and kind of expanding the keyhole, right? And we're not looking through the keyhole no more. We're starting to know there's more beyond the wall, right? And how we kind of go about um, facilitating care and whatnot. Like we can do better, in my opinion. I always do better. And I want to say too, while I'm on the air here, a lot of people say I come off a little harsh, and that's okay. I'm hardest on myself. And so that's where that comes from. And nothing I do is good enough. Everything I do could be better. I beat myself up all the time. At the end of the day, I got to sleep at night. And so that's why I'm always challenging myself to do better for my spiders and my animals under my care. And so, you know, I kind of this expectation for my peers to do better is just there. It's not like I'm trying to be a jerk. It's just like, let's just all do better. You know what I mean? Because we can.
yeah,
But Becky, you're so good at talking and explaining these things to people, and you think of things that I don't even think about, and I think it's very valuable. And I hope that you continue to take the time to talk to folks and other content creators, give you a platform to speak and keep going because it's very important. And you're going to learn more, and you're gonna find things out. And I hope you share that with us because we're hungry for it and we need it. So thank
absolutely.
you. Yep. I appreciate that. So, anything else, Becky? You want to wrap it up? We're a little over an hour now.
No, I could go on little nitpicky kinds of fun little science things all
Same.
day. So
Yep.
I'll let us wrap it up here.
Okay, I think we can put a bow on it. Guys, links are in the description. I'm gonna have Jessica, as soon as I'm done with this, hop on and make sure everything, she's looking at me, make sure everything is where it's supposed to be. Check out Becky's blog. That's on the banner here. Girl with Darwin Tattoo. More martial arachnids, martialarachnids.com. Boom, there it is. I love that, by the way. Okay, Becky, stick around. We're going to talk a little bit. I'm going to drop some names and kind of see how you feel about that. Everyone else, guys, thank you so much. I hope we have more conversations like this in the future. We love you guys. I love you guys. Thank you so much. See ya.